Well, let me tell you very briefly about today's talk based on yesterday's talk. Yesterday's talk was very briefly going into this very important fact that we have bought a belief system called genetic determinism, the belief that genes control our biology and control our lives. And we bought this based on a premise that was actually founded by Charles Darwin in 1859, that hereditary traits control life. And then there was a search for what those hereditary traits are, and it led to the discovery of the DNA double helix, and everybody just bought into, oh my God, this is exactly what we were looking for, and that genes control our lives. What my research that I talked about yesterday was that this is completely wrong assumption. In fact, it's so wrong, it's actually quite disturbing as a consequence of medicine is based on that. And by trying to understand all of our ills through genes, we have gone in the wrong direction. That in fact, genes do not control life. That life is controlled by perception. And this is what yesterday's lecture was all about, how what we see in the world is converted by the cell into both behavior and to genetic expression. So basically, what we were getting at was our whole fundamental belief system about genes is corrupt, and that we really have to understand a new way of looking at life. Then I started to follow through and talk about how we acquire perceptions and how our lives are really run by these hidden perceptions that we got, especially between in utero development and the first 6 years of life. And then the conclusion was, of course, that if we can understand that programming, that time period, and adjust for it, we can change our biology as of the day. Right now, we can change it. So I come in today's lecture with a continuation of that story, but as it relates to the story of evolution. And as we talk about the story of evolution, we're all familiar with the stuff that's in the textbook about the concept called neo-Darwinian evolution. Darwinian evolution was the philosophical understanding about the survival in the struggle for existence. The neo part added to the front is an extension of Darwin's work using the data gained from molecular and cellular biology about the nature of the molecules of DNA and RNA, et cetera, like that. I could summarize very briefly that there are two fundamental steps in the theory of Darwin's theory of evolution. And basically, and it said this: genes control life, and that's, of course, the first step. And then, as a consequence of that, alterations of the genetic message occur, and they occur randomly. And this is a very important point because it suggests that all mutations are just accidents, that in copying the genetic material, that an error is occurring during the copying. You put in the wrong bases, and then you change the blueprint that the DNA represents, and then the product of that gene is altered. And then as a result of that, you either cause a disturbance to the system, or in the most extreme and unlikely situation, an alteration could actually improve the situation, okay? But the whole premise is that it was random. As a result of that belief, it says, then evolution was just a whole series of accidents. And when we look at it that way, then it says, how did we get here? And it's just like, it's just a bunch of accidents. So where are we going? Well, we have no idea where the heck we're going because we don't know what the next accident is going to be. And so essentially, we live from day to day with a surprise of what we might expect for tomorrow. In the process of having a random mutation, as I said, sometimes the product of the gene that's mutated enhances life, which is very infrequent, not many studies that show that. And almost all of the time, a mutation actually takes away from the viability or healthiness of an individual. When we talk about natural selection, what this point is that nature will select the weakest elements to drop out of the picture so they don't propagate those weak elements in the population. So nature selects by eliminating the weakest. Now, it's a very important, it's that story of whether the glass is half full or the glass is half empty. When you talk about nature selecting the strongest, or nature selecting the weakest, in the sense of nature selecting the strongest says, oh, the strongest ones are propagated by nature. No, nature doesn't really care about the strongest ones. Nature eliminates the weakest ones. There's been this great misunderstanding that all of evolution is driven by the strongest element in the group. And there's a wonderful paper by Timothy Lipton that was in Nature and said, oh my God, would we make a mistake? You know why? Nature doesn't give a damn about the strongest in the group. Nature cares about what the whole group does in regard to the interaction in the environment. So it doesn't care about the individual, but it cares about the whole group. And that becomes important because we focus on the individual when it's really the action of the group that nature really considers. So this is our belief system. Evolution based on randomness and probability and just chance, and nobody knows where we're going with it, okay? I'm going to tell you, I don't support that. It doesn't work, and there are so many reasons as to why biologically this doesn't work that we're really in search of another theory of evolution. And now why this is important is because people hold on to the Darwin theory with such, they hold so dear that if anybody challenges the Darwinian theory, they're almost like called creationists or something else, and they get all yelled at. Well, I'm actually not a creationist, but I will tell you something that through the new understanding, I actually believe that evolution and creationism are simultaneous events, and that there was not one or the other. And that why this is important is because it also tells us if there's a direction we're going in, can we know where we're going? And why is that important, of course, is because our survival is being challenged right now. Right now, we are deep into the 6th mass extinction to hit this planet. There have been 5 previous mass extinctions on this planet that nearly wiped out life, and in every case, just took it down to a nubbin, and then all new life started from that kernel and started all over again. The previous 5 mass extinctions are all attributed to extraterrestrial causes, such as an asteroid or comet hitting the Earth. The 6th, the one we're in right now, is unique because the cause is us. We are destroying the environment that we live in, not owning a simple reality. We are made in the image of the environment. So as we destroy the environment, we destroy ourselves in the entire process. I'd like to offer something brand new based on my science on how the cells work. Very briefly, I talked about it yesterday, so this is just a quick review. What are cells in regard to us? Number one, cells are what you're made out of. That when you think of yourself as a singular entity, that's a grave mistake, because the truth is that the living things that make you up are the cells. You represent the communal action of 50 trillion individual living entities that make you up. So when you see yourself as a singular, is really a mistake because you're a community. That those 50 trillion cells are very wonderful in this regard because they have all the powers that you have in the sense of your human physiology. Everything that you can find in a human physiology book is already present in every cell. Every cell has a digestive, respiratory, excretory, nervous system, immune system, reproduction system. And the simple fact is, you can only do what a cell can do. In other words, you can't carry out a function that cells can't do because you're made out of cells. So that puts us back into a ground level of, when I talk about cells, I'm really talking about you. And as you'll see the connection at the end, there's a great affinity of seeing this as a connection that's important for our awareness. So how do cells work? I brought up the concept that cells are machines. In a sense, they're made out of proteins. The proteins are the building blocks. There are about 200,000 different forms of proteins that make up a human. That the proteins interact with each other like cogged gears. They interact and they move, and in that movement, they generate the functions of life. That the collection of all the proteins is called a proteome, and this is science's next endeavor after the genome to find out and identify and list all of the proteins that make up the system. I gave yesterday an example of a protein. I used calmodulin. I talked about, well, what gives the character to a protein? I said, underneath that exterior is a backbone. That all proteins are linear strings of amino acids. That the shape of the protein is due to the relationship and adjustments of energy between the amino acids. So some amino acids are positive and some are negative. And so when this backbone is built, then it will adjust itself so that all the charges are essentially neutralized and balanced as best it possibly can. And then I said, but where does life come from? And life is not just having the physical parts, life is actually having the movement. So movement is what's generating the movement becomes relatively important, because how can a protein move? And the answer is this. I said that the backbone configuration or conformation is based on the distribution of electrical charges. So therefore, if you change the charges in that protein, when you change the charge, then the backbone will adjust itself to accommodate the new charges. The adjustment of the backbone generates movement, just like this. So proteins are like little miniature people. They move. And as a result, the movement that they represent is turned into behavior and biology. So the summary statement was, proteins make us up. that the proteins respond to signals from the environment, that our conventional understanding of the signals from the field that influences conventional medicine says only molecules and substance of chemistry can cause the proteins to change their backbone. But we now know that that's not true, that energy fields, especially in the quantum mechanical range, those kinds of energy fields are called constructive and destructive interference. Those energies can actually cause the backbone of the protein to change, so that when these signals bind to a protein, it generates behavior. Behavior is the expression of life. Okay? Now the question is, well, what controls the signals? Because in this case, the proteins exist, but the signals are the regulatory element. When a signal is present, the protein works. When a signal is not present, it may not work, or vice versa. So the question is, what controls the cell? And that's where we came in yesterday. We talked about the brain. I said it was the membrane. The membrane, the skin of the cell, that the genes do not control the cell simply by telling you this. I can remove the genes from the cell, and the cell still has its life, still with the same behaviors and ability to respond to the environment. So the genes, in their absence, suggest that they can't be the ones that's controlling life. We come to the understanding that the skin of the cell is actually the brain of the cell, and I mentioned yesterday that the human skin is also the derivative of the human brain, so our brains are also derived from the skin. The relevance about that is the skin is the interface between the environmental signals and the proteins that create life. So when the signal comes to the membrane, it hits the membrane. There's a switch at the membrane which will interpret the signal and then send a secondary signal to generate the behavior. So the switch has two parts. One part reads the primary environmental signal, and the other part of the switch is what sends the second signal, or second message in some cases. That's what it's called. So the issue is, let's take a look at this membrane, because we're going to find something very interesting about this. And it works like this. The membrane is this structure. It's a bilayer structure. It's actually two rows of molecules with a mirror image of each other. So I'll give an example. There in the middle is the head of a molecule, and this is called a phospholipid. The head is polar, meaning it has positive and negative charges. But the legs of the molecule are nonpolar. They're oil. And so the difference is, it's like oil and water. The heads absorb and bind to water, but the legs are oil, and they resist water. As a result, the membrane makes a fine barrier, because whatever is in the outside environment, generally dissolved in water, is generally not able to pass through the membrane because of the oil phase in the center, and that's why the membrane's a barrier. So when I want to show you an electron microscope picture, it's almost an exact image of this. Here's a picture of the human cell membrane, and what you can see is the middle, there's a dark, light, dark layering. And if I go back one slide, you can see there's dark, light, and dark. And so the model and the image are pretty much the same as each other. Now, as I said, the middle part is oil-loving. So if I actually cut out a piece of this membrane right here, it would be like a bread and butter sandwich, where the heads of the molecules that love water are like bread, because bread soaks up water. And then the middle of the sandwich, butter, would be the equivalent of the inside. So here's an example of what the cell membrane looks like. And the relevance about that is that this is the lipid leg region, and this is the phosphate head region. And why is this important? Because imagine we're in a cell right now, and that over our head is a membrane, a big bread and butter sandwich, and I'm sitting here saying, well, let's say we're thirsty and we want the universe to give us something to drink. So I ask the universe, would you give us something to drink? And so the universe dollops on some grape juice. And the issue is, okay, there's been a big dollop of grape juice thrown on the ceiling here, which is this bread and butter sandwich. Will we or will we not be able to drink any of that grape juice? Yes or no? No. And the reason? The lipid won't let it get through. Well, I can show you if I section the sandwich. Let's section it. As you can see, yes, as soon as the grape juice hits the lipid layer, boom, it's a barrier. It can't go across. Well, that suggests then something very interesting about the cell is then how does the cell get food inside, and how does it get waste out? How does it communicate? And the answer is because the membrane has more than just the phospholipids. The membrane also has proteins. Remember those that were here yesterday, I showed a poppet bead necklace, and I said that the poppet beads were amino acids in the backbone of the protein. Well, some of the amino acids are oil-loving amino acids. So when you have a region of oil-loving amino acids, they prefer to be stuck in the butter part of the sandwich, and the hydrophilic or water-loving parts of the protein extend above and below the membrane. So these proteins are anchored into the membrane. They're called integral membrane proteins, IMP. The integral membrane protein. Sometimes there are multiple regions, the parts that extend out of the surface have antennas on them. Generally, they're sugar antennas. And these antennas are part of the tuning mechanism of the protein, like tuning forks. So that these proteins are like tuning forks that resonate with environmental signals. So when an appropriate signal comes up that complements a tuning fork, it causes the tuning fork to respond and causes the protein to change shape, and that's the signal binding to the protein. As I said, sometimes there are multiple regions where there are lots of lipid accumulations in the protein chain, so the same chain can weave in and out of the membrane. So in a sense, if we looked at a membrane like this, the proteins would look like olives stuck in our bread and butter sandwich. So there they are right there, the proteins in the membrane. And the relevance about this now is, okay, now I go back and I say, okay, we have a bread and butter and olive sandwich for the ceiling. And then I ask the universe, would you please give us that grape juice I asked for? Now the question is this, will I or will I not, with an olive sandwich, be able to drink the grape juice, yes or no? Yes. And how did the grape juice come through? Through the olives. Yeah, but the pimento is in the olive, right? Yeah, but the pimento is blocking the channel, right? Let's take a look at it. Oh, you see, when the pimento is in, then nothing can get through the olive. But when the pimento is out, the olive creates a channel. And as a result, I can let things in. Well, the difference is, what does it mean when a pimento is in or out of the olive? And the answer is, if you were here yesterday, I wish you all were. The point is, proteins change shape. So I could have a protein that's tightly wrapped, and when a signal comes in, the backbone of the protein relaxes, and as it relaxes, it opens up. And when the signal's gone, it closes again. So I can open and close the gate. So far, so good, right? Okay, well, the next thing then we talk about is, well, what are these proteins that are built in the membrane? I said there are two classes, one with antennas, which are called receptors, and they're like your receptors, which are in your skin as well. Eyes, ears, nose, taste, touch, pain, pressure, they're receptors built into your membrane. The second class of proteins are effectors, because I said the switch has two functions. One, to receive the primary environmental signal, and the second is to send a signal into the cell to engage the protein machinery of the cell. So the switch has two parts, a receptor and an effector. So the issue about a primary signal picked up by the receptor, the secondary signal that goes into the cell and controls the protein gears that do life, is the concept of what the effector is actually doing. Okay, just to illustrate this in a dynamic form, I showed it yesterday. We'll do it one more time, and it shows like this. Here's the two components, the antenna on the receptor, scanning the environment, looking for a signal, the connector between the receptor and the effector. And again, look at the shape of this. It doesn't combine or match the shape of this until the signal comes. When the signal comes, it changes the shape, causing this protein to couple to this protein. And when that happens, this protein, like the olive, opens up and sends a signal into the cell that controls the nature of whatever functions attached to that signal. When the primary signal is gone, the switch disconnects. Okay, now the irrelevance about all this is this represents an important switch. This is a switch that interacts between the environmental signal and the function of the cell. This is the elemental unit of awareness, because these two proteins together are required for a cell to respond to an environmental stimulus. The first part, the receptor, which is an awareness of the environment, is reading the primary signal. The second protein is sending a physical sensation into the cell. Together, they form a unit by definition, which is an awareness of the environment through physical sensation, which is called perception. That is a molecular unit that translates or transduces an environmental signal into behavior. So anything you're doing when you're responding to the environment, you must have perception units that are reading that environmental stimulus, whatever it is, and then adjusting your biology in response to that stimulus, okay? Now, back in 1985, when I was putting all this understanding of how the brain of the cell worked, the membrane, what really clued me in and drove me, like, wow, just changed my life was I wrote the definition of the membrane down in the functional definitions I just described. And I sat there in 1985, and what did I write? Well, the first thing is I characterized, remember the lipids that were making up the bread and butter sandwich? It's a bilayer, that it's a mirror image of each other. Note that the molecules are lined up like soldiers on parade. They're in a crystal form. When molecules have a regimented orientation, they're called crystals. But what's really important is there are no chemical bonds anchoring these next to each other. So all the molecules are just standing next to each other. They're not connected to each other. Imagine a lot of people in an elevator. Everybody's standing tight like this. And somebody in the back of the elevator wants to get out. So what do? They all move like this, and somebody gets out to the front. What was the point? All the units in the elevator were all in registration, but they were also movable, that they weren't connected to each other, and they moved by just shuttling around. Well, the point is they're crystalline, but they're not like salt or diamond or quartz. They're movable crystals. So it's called a liquid crystal. So the foundation of the membrane is called a liquid crystal. The next thing I said, well, about the communication. I said, remember when I just put the blue stain on when it was a bread and butter sandwich? It's a non-conductor. Nothing goes through. Then I said, well, wait, put the olives in there. And then I said, yes, now stuff can go through the olives, but here's what's important. When the olives are in there, they don't let everything through. There are potassium channels, sodium channels, chlorine channels. Okay, these are different channels, different olives. And what's the result? They only let in the certain things. So all of a sudden it says things can go through, but not everything. So somewhere it's not a non-conductor. It's not a full conductor. It's in the middle. It's called a semiconductor. So I say, okay, membrane's a semiconductor. And then the last thing I said, well, what are the fundamental proteins that make up this awareness? And I said, well, there were two of them. They were what? The receptor and the effector. Well, the effector I've been talking about is a channel. So basically I said, well, there are receptors and channels. But there's a synonym for receptor called gate. So I write down that the membrane contains gates and channels. So I'm sitting there at that moment in 1985, sit back and look at what I just wrote. The membrane is a liquid crystal semiconductor with gates and channels. And all of a sudden I stop and I think, yeah, wow, you know, I just read that definition someplace, but it wasn't in the sense of biology. Where the heck did I read that? And I look, and there in the corner of my desk, my first Macintosh, and right next to it is a book from Radio Shack called Understanding Your Microprocessor, a book for simpletons like myself on how a computer worked. And I said, oh my goodness, I opened up the book, page 3, introduction. And it has a definition that says, a computer chip is a crystal semiconductor with gates and channels. And I go, wow, what a coincidence. What a coincidence. The computer chip and the cell membrane have the same definition. And then all of a sudden, I started to get a little deeper, and I realized, oh my God, this isn't a coincidence. These are exactly the same. Structurally and functionally, they're exactly the same. And so I started to look at it and I said, well, what's a chip? Well, a chip has a base that's a non-conductor right here. Look what the name of it is. It's called a channel. But most of the times the channel is not open. There's no conduction of the electricity from the negative to the positive right here. This structure is called the gate. And what happens is this is the equivalent of the receptor. When I activate the gate, it causes the channel to become a conductor. Well, remember the protein? When the receptor wasn't activated, the protein was closed. When the receptor was activated, the protein was open. And so basically, it says that, my goodness, when you activate the gate, it causes a change in the conduction property of the channel. And when the channel is conducting, the electricity goes through the channel, and you have a passage or flow of current through it. And this is exactly how the chip works. And when the gate is shut off, then the flow stops again. Well, isn't that exactly what I said happened here? Here's a receptor, here's a channel. When they're not connected, the channel is closed. Important point. Geez, I should have mentioned this, but I will now. Inside the cell membrane, there's a negative charge. Outside the cell membrane, there's a positive charge. And this is called the membrane potential, that all cells are batteries, and that they function by having this charge differential, so that between the membranes, positive on the outside, negative on the inside. Yeah, but that would say then if I opened up the channel, what would happen? And the answer is, if I couple the receptor when it receives a signal to the channel opens up and the current flows across. Is this or is this not exactly what you just saw in the other chip? And the answer is, it's exactly the same. And what really freaked me is like, oh my God, the genes don't control biology. I already knew that. I've been working 10 years on understanding how the membrane worked. Now I realize this, that the cell is a programmable chip, that the nucleus is a hard disk, and the genes are programs. But like in your own computer, the disk doesn't drive your machinery. You have to drive the machinery. And so basically the membrane is the interface where the environmental signals, when typed on the surface of the cell, engage the functions out of the hard drive. And so when you put this all together, you start to recognize the cell membrane's a chip. Well, I did that in 1985, and in 1997, this paper came out in, oops, go backwards. This paper came out in Nature, a biosensor that uses ion channel switches. And here's a cell membrane. Here's a receptor. Here's a signal binding to a receptor. Underneath the cell membrane, they took a cell membrane and they stuck it to a gold foil, a piece of gold foil like an electrode. They put an electrolyte solution between the membrane and the gold foil. When the channel opened after the receptor was activated, the flow of electricity through the channel activates the electrode, the gold foil, and then they get a digital readout of the action of the receptor. It says right there, the results are displayed on a handheld digital screen. Meaning what? They created a chip out of the cell membrane. But in truth, they didn't create the chip. That was God created the chip. They just stuck the gold foil to the bottom and were able to get the digital readout. So the point is, what my idea was a hypothesis. What this is technology based on that understanding itself. So what they've demonstrated is the cell membrane doesn't just look like a chip. The cell membrane is a chip, and this is very important. I'm not saying it's an analogy. I'm not saying, hey, the cell membrane is like a chip. No, no. The cell membrane is a chip. It's an organic chip. And what does that mean? Then we go back to the cell and recognize that a cell and a computer are the same in regard to the components of computation. That this is an organic, this is an organic chip on the right, and this is a silicon chip, carbon chip, silicon chip, okay? But the components that are in here are the same as the components in here. I can find them for you and tell you what they are. So when I look at the computer on the left, the one we're all familiar with, I say that central processing unit, the box that was normally under the monitor, is the what? The part that takes the data and converts it into different information. Yeah, that part is the cell membrane. The cell membrane picks up environmental data, light, sound, smell, glucose molecules, estrogen molecules, whatever that is, and converts it into biological awareness. So the membrane is the central processor. And then I ask, okay, how do you put data into this computer on the left? And the answer is you type on the keyboard. So data is entered via the keyboard. And I go, oh, I forgot, I'm so ahead of myself here. To go back to that central processing unit, what we talk about are bits of data. Your processor handles bits of data. A bit, by its definition in the electrical engineering, is an I/O. I means input/output. A unit that has input/output is a bit. Then I say, yeah, but I can tell you where that is in the cell membrane because the receptor is the input and the effector is the output. That is a bit of data. So basically, it says, yeah, it's exactly the same. It's an organic bit of data. Now, going back and saying, well, how do you type the data in? And the answer is the cell surface is the keyboard. All those antennas sticking up are the keys on a large keyboard. That a certain chemical will come, will select a certain key. An estrogen molecule doesn't hit a histamine receptor. An estrogen molecule hits an estrogen receptor. So the entire keyboard is what the keys are that respond to the environment. So the surface of the cell is a giant keyboard. And as the cell moves through the world, whatever is out there, the information is being typed on the keys. And as the information is being typed on the keys, it causes a program to be engaged inside in the exact same manner that when you type on your keyboard on your computer, you're engaging the system inside. The nucleus, as I mentioned, turns out to be a disk. It's just the programs. That's why you can do, as I said, the enucleation experiment. You can remove the nucleus from a cell, and it doesn't change the behavior, because once the information is downloaded, you don't need the disk. So if I give you a disk for your computer with a program on it, you put it in your computer, you load the program, you can eject the disk. And guess what? You can still control the program. The genes don't control it. The disk doesn't control it. So the genes are just the disk. And then when you put all this together, you recognize, well, you see the data on the screen. And here's the beautiful part. The 3D image of the cell is the screen. The 3D image of your body is the screen. This data of information that you respond to is expressed on your screen. And that's what diagnosis is about in medicine. I can look at your screen and tell you about what's being programmed. So basically, how you are, your health or your disease states, are readouts of the program. That's exactly where that comes from, okay? There are different controls for men and women on this particular... But now comes, I think, for me, what is perhaps for me, now I hope it is for some of you, one of the most profound pieces of information that I ever saw in my life, and that is this. This represents a bit of data. That when we talk about the power of a computer, we talk about the data handling capacity. How many bits of data does your computer handle? And what we do is we get bigger and more powerful data processors with more bits of handling power, more bits. So it says that the evolution of the computer is what? Increasing the data handling capacity, right? More bits. Okay, then I say, well, yeah, but these are the bits. And here comes this now beautiful piece of twist of nature, of beauty. And it goes like this. When I look at the cell membrane, remember the lipid bilayer with the lipids, the phospholipid heads and the tails? Those molecules have an exact size. And they have to stand tail to tail. So there was a bilayer. Remember, heads and tails up, heads and tails meeting in the middle. That means that the diameter of the membrane is of uniform thickness. You got that? All the way across. It's the same for bacteria, same for humans, all through evolution. The reason is this. The molecules determine the thickness. Well, here's the point. The proteins that embed in the cell membrane, this is a large picture, but are about exactly the same size as the membrane itself. The proteins are about the same size. Now listen, remember I put olives on a sandwich? And I said, the more olives I have on a sandwich means what? More bits, right? More awareness. But I said, wait, I can't stack the olives on top of each other. Why? Because the membrane has a uniform thickness. This is a real important point. You got this? If I want to add more olives, I can't add it up and down. Where can I add more olives? Sideways, left and right. What does that translate into? You want more olives, it goes like this. How many olives can I put on a piece of cocktail rye? And how many olives can I put on a big slab of pumpernickel? The bigger the bread, the more olives, right? Awareness is what evolution is based on. Evolution is the expansion of awareness. Yeah, but awareness is olives in a sandwich. Guess what? For the first time, we have a mathematical construct to model evolution. What? More membrane surface, more awareness. Why? More membranes, more olives. And all of a sudden it says, oh my God, evolution has a mathematical correlation to it. It's not a random event. You can model evolution using geometry. Well, what geometry would you use? Well, I want to tell you, this is on the mathematics of human life, and my dear friend Ralph Abraham, I hope he's not in here because he's like the master of chaos theory, and I hope I'm not embarrassing myself in front of him. He's not here. Cool. Ralph Abraham talks about fractals and chaos, and I want to show you this little excerpt, so it's not me just saying it. It's an excerpt out of this article on the mathematics of human life. Mathematical studies of fractals, this is geometry I'll talk about, reveal that the repetitive branching within branching structure of a fractal represents the best way to get the most surface area within a three-dimensional space. So if I wanted to take a membrane, which is a two-dimensional surface area, and put it into a three-dimensional space, what geometry would I use? Fractal geometry. And then all of a sudden you say, well, what the heck is fractal geometry? Well, we'll go back and say, you and I took geometry in school, but it was called Euclidean geometry. Well, what's Euclidean geometry? Well, remember the graph paper? And you put cubes and cones and spheres and all those things. You plotted them out on a graph paper. Well, when we plotted them, we were using Euclidean geometry. But let's talk, how many dimensions do these structures at the bottom have? When we talk dimensions, how many do they have? 3 dimensions. This is a flat surface. How many dimensions does that have? 2. There's a line. Guess how many dimensions that has? 1. And there's a dot. Okay, you can guess. How many dimensions in a dot? 0. Isn't that interesting that the geometry that we're working with has whole dimensions? 0, 1, 2, 3. And the issue is, can I have somebody have a blank piece of paper that I can just destroy? Just a piece of paper, I need a destruction. I got one thing. Oh, so small, so bigger. I have a big audience. How many dimensions? Not counting thickness. How many dimensions? Okay, watch. Sleight of hand. How many dimensions? I don't know. 2.5. Why is it 2.5? And the answer is, well, it's actually not 2, and it's not 3. I'll tell you why it's not 3. If you section this anywhere, there is no three-dimensionality to it. It's all just 2 dimensions folded in what appears to be 3. So it compensates and says, it's not 2 and it's not 3. It's a fraction of a dimension. Well, we don't have that. There is no fraction of a dimension in our Euclidean geometry. And here's something else that's unique. Make a tree using Euclidean geometry. You did when you were 5 years old. What did you do? You took a cylinder for the trunk and a ball for the top. But that's not nature. Make nature using Euclidean geometry. Make a cloud. Make a mountain. Make a fern. Nope. You know what? Interesting fact. Nature that we live in is not, programmable using Euclidean geometry. That geometry does not map nature. Nature responds to a different geometry. What does it respond to? A fractional geometry, fractal geometry. What's fractals? Well, they're beautiful equations, and Dr. Kaku yesterday in physics talked about what? He said that when we really understand how life works, the equation is going to be a little tiny, short, simple equation. Well, guess what? The fractal geometry is the simplest mathematical equation. All it has is multiplication and addition. That's all it is. And it's very short. And what is it? It's an iterated equation. What does that mean? It repeats itself. And how do you repeat an equation? The answer is this. You put in a number, start the equation, you get a result. You take that result, feed it back into the same equation, do it again, and you get another result. Take that result and feed it back in over and over and over and over again. I'll give you the simple one. Take a line and cut it in half. That's called the equation. So I start with a line this big, and I cut it in half. Boom. And now what do? Is I take one of those lines, half, put it back in the equation. What does the equation say? Take a line, cut it in half. So I take that one, cut it in half. Now it's this big. And I put that half back in the equation. I cut it in half. How many times can I do that? Infinite. And this is the character of fractal geometry. It's an infinite geometry. And how does it work? Well, I'll give you an example of an iterated equation. This is called a Koch snowflake. It's an equilateral triangle. And the equation says, on every free surface, create an equilateral triangle whose dimensions are the length of that surface. So the next round is the orange triangle. This equilateral triangle has the length of this side. And same on this side and this side. But now look, I have new sides. I have this side, and I have this side, and this side, and that side. So I make a new triangle, and that's the yellow one. But then on the surface of the yellow one, you see what I'm going through? How many times can I do that? Infinite. Okay. Now, this is the fractal equation, and it's basically a very simple equation, and it deals with taking the number, putting it back in. This is called the Mandelbrot fractal after Benoit Mandelbrot, who worked this at IBM. Actually, he didn't come up with the concept of fractals. Fractals were at the turn of the last century, 2 centuries ago now, by a mathematician called Julia. And the issue is, to see the result of the fractal equation, it's an iterated equation. You have to do it millions of times. You have to take the same equation and repeat it and plot it and plead it and plot it and repeat it and plot it a million times. They didn't have that many graduate students. So the reality is Julia's work was theoretical because nobody could sit there that long and run a million or more equations until Mandelbrot did it at IBM with a supercomputer and then created the first image of a fractal, and this is it. And basically what you're looking at is not a triangle, but in this case a circle, and then you put the next generation is, and you can see the generations in color. So the first one is dark blue, the second one is the green, the third is light blue, the fourth is red, and as you start to see, you go deeper and deeper. Well, blow this up. What is all this fuzzy-looking stuff here? Blow it up, and there you see it again. And what do you see? The image of the Mandelbrot repeats itself ever, 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 ever deeper and deeper. And all that white fuzzy stuff, magnify that, and guess what's in there? The same Mandelbrots all over and over again. Now, you take the Mandelbrot set. Here's an example of it plotted out. And what's interesting is it's sitting on a large circle right down here, and then there are other ones on top, and other ones on top, and then you see all this stuff, and you say, what the heck is that? And the answer is you blow it up, and you'll see chaos. And in between those chaos, if those are up in the front, you can see the image repeats itself right through the whole structure. You can see it repeating itself again and again. So an inherent character of a fractal is it repeats itself. But number two, you can add chaos. What is adding chaos? Well, remember I said, let's say make the triangle the Koch snowflake, and I say put the triangle in the dead center of the face. But now I can add chaos. What I can do is add part to the equation and say flip a coin. If it comes up head, move it to the left of the center. If it comes up tail, move it to the right center. I add a little variation. And when you do that, you can take the same Mandelbrot equation, add the variation, and look what you get. And all this stuff, you say, what the heck is all this stuff? Blow it up. And it's more in designs that are just phenomenal in nature. And you can go deeper and deeper, and it repeats itself again and again. And so what it says is that as you go through the geometry, higher or lower, the same images, or self-similar images actually, are repeated throughout the spectrum of infinity. Okay? One more, let's see. Let me show you this. This is a fly-through. It's not the greatest resolution, but you're flying, imagine you had crosshairs in the center, and you're flying through a fractal. And this is what the fractal will look like as you fly through it. As you start to go deeper and deeper, it gets more exotic. And oh, look, here's the Mandelbrot set. But we're going right here. We're focusing right here. And just keep your eyes right there, and we'll come back deeper and deeper and deeper. And look at all this elaborate, beautiful symmetry and stuff that's going on here. It's all coming up. All this geometry is what? Here it comes again. The Mandelbrot set. And it says all this geometry is variations on the chaos of that Mandelbrot equation. Okay? Now, I say, let's look at nature. Well, here's a picture of mountains and clouds. You know what? This is a fractal equation. This is a computer-generated picture by putting math into a fractal equation. And boy, does that look like the Earth or not. You want to see something else? There is a fractal equation. And what we found is that the Earth and the biosphere are fractal images that are repeated infinitely above and below. Here's an example of a real vegetable. It's sort of a mixture between broccoli and cauliflower. And look at it. It's a fractal. A perfect, beautiful fractal. And then, here's my backyard. And this is a tree. And you say, is that a fractal? I can tell you it is. Why? Watch this. I will take a piece of this picture, cut it out over here, that square, cut that image out, and I'm going to move a copy of that image, and I'll just bring it up into the tree and drop it, paste it, cut and paste. Paste, remove the lines. Where is it? Self-similar. The tree is a self-similar repeating image. So I can take a piece of the tree and it represents the tree anywhere else. Nature is fractal, guaranteed. Okay, that's what's going on. Relevance of fractal. As you go up or down the infinite scale, the images repeat themselves. And this is this is called the Rosetta Stone, found in Alexandria, Egypt, 1799. When they found this stone, they had no understanding of all the hieroglyphics all over the ancient buildings of Egypt. There was no understanding about it. But when they found this stone, which is about 3 feet high, 2 feet wide, there were 3 languages on it. Greek at the bottom, Demotic, which is a more modern form of Egyptian writing, and the ancient form hieroglyphics. When they interpreted the Greek story, it was about some kings and queens. Then they went up to the Demotic, and they started to interpret that. Guess what? Same story, same kings and queens. The intuitive leap. If the bottom 2 tell the same story, what's the possibility the top one is telling the same story? And through this little piece of hieroglyphics, they deciphered the hieroglyphics in all of Egypt. And it was all based on what? It's based on, oh, as it is above, it is below. Why is that relevant for the story that we want to have? Because when I look at the Rosetta Stone in regard to biology, there's a cell, there's a human, and there's a human civilization. I told you already that you're made in the image of a cell. Everything that you do is built into the cell. You are a cell reincarnate. But now we, you, me, we are cells in a larger organization called humanity. Relevance? If you understand the principles of life at the lower level, then you fundamentally know the principles of life required at the higher level. And why do we need this now? Because we are destroying our world because we have no plan. And if I wanted to know what the plan is for life, I can tell you exactly what the plan is for life. No guessing. Look inside your human body. You got 50 trillion citizens. Each cell is an independent living entity sharing one environment. And when you're in health, can you imagine how many individuals that are in that little tiny space? Compared to the number of people on this planet, that makes them look insignificant. And yet they live in harmony. And they're able to share, work together. They have energy exchanges. They exchange an economy of ATP molecules. They support each other. They take care of each other. You want to know how to take care of ourselves and live in the world? Study how cells live in a human cell body. Because all the rules for living there are what we need to live here. And so that the mission of life is not to go through the concept that we live in a random world and God knows what's going to happen tomorrow. I can tell you what's going to happen. Why? The images repeat themselves over time. So when you look at higher or lower, it doesn't make any difference. I'll give you an example. We talk about particles and waves. Particles and waves. That the waves on the left are nonlinear. The particles are called linear. They're structured. And while everyone talks about, let me talk about the waves. No, I want to talk about the particles. Everybody's missing the picture. You know where the action is? At the interface, where the wave hits the particle. That's a chaos zone. That's where the waves and the particles interact, and there's an etching at the surface of the energy of the waves and the physical energy of the particle. And this is a chaos place. And it's the same whether the particles are big or small. The Earth is a particle. The waves of the solar system are the nonlinear waves. And the chaos is where? At the surface of the Earth. And the interesting part about that is what happens at the surface of the Earth. Well, think about this. Before there was life, what kind of chemistry did we have? Inorganic chemistry. Non-life. The kind of chemistry that seeks stability. A rock likes to be a rock. It doesn't want to change. It'll stay a rock for as long as it possibly can. But when pigments evolved, pigments were able to trap photons of light from the sun, the waves, and take that energy of the photon and use it to move electrons, and in the process take inorganic chemistry and create organic chemistry. Yeah, but we're made out of organic chemistry. So what does it say we're actually made out of? And the answer is this. We're made out of the earth and the sun. We are both at the same time. We are energized earth. An energized earth does not want to sit still. Energized earth is an active, moving, organic matrix. And so we are life, but we represent energy, half, matter, half, coming together. And so when we look at life, if we look at the earth before there was any life here, photons would hit the earth, be absorbed, and then the energy would radiate off as heat. But once that inorganic chemistry was endowed with the ability to express organic chemistry, it changed. But in this case, let's just watch what happens here. Organic chemistry is the one that seeks activity, okay? Now watch. The photons, oh, it's not going to happen. I'm sorry. It was an animation that didn't seem to play right now. What was the point? Photons of light came down, hit the earth, and after the evolution of organic chemistry, the photons didn't necessarily radiate back away again. The photons were now trapped in a matrix of organic chemistry. So the sunlight is accumulating on the surface of the earth. The earth is energized at the surface. And the waves come from the sun and the solar system. The particles come from the earth. And the chaos zone in the middle is where life occurs. So when we look at it, we can then relate and say, as is above, as is below. Then if we understand it this way, we physically came from the planet, but we energetically came from the sun. And in looking at that, then you could call the planet Mother Earth, because this is the physical origin of where we came from, and Father Sun are the waves. And then even in our own reproduction, the egg is the earth that we come from, and the sperm doesn't deliver any earth, it just delivers information. So the reality is, it's still the same fractal imagery, I don't care what level you want to look at. But then what we want to do is not run out of time. So I'll quickly show you how evolution, I see it how it occurred, is that first in the primeval seas, at first, all there were, oops, go back. At first, there were just proteins floating around in a solution. That these proteins are just responding to the environment and moving all around. But the phospholipids are self-assembling structures. A membrane is a self-assembling process. You can shake them up and they create membranes automatically. The proteins that get trapped in the membrane are in a separate domain because of the lipid layer. If the environment changes on the outside, the environment's still stable on the inside. So life occurs within a membrane structure because those are regulatable domains. And so life needed the membrane first to start. But now I have proteins that can carry out a function inside the cell, and cells started to develop into communities that, so the bacteria cell was not a single cell as we know it. They live in a community and interact with each other. But they start to come together in what is called a biofilm. And a biofilm represents a collection of bacteria that surround themselves with like a membranous structure. And that within that structure, the bacteria exchange information, even including DNA. And there are different bacteria so that you can take a bacterial population, put it in a membrane. But if you do that at some point, then those bacteria in a membrane ultimately evolve into the more advanced cell called a eukaryotic cell, where the structures on the inside appear to have been formerly bacteria surrounded by a common membrane. Then you take that human cell, which is the fundamental unit that we're made out of. Cells started to come together in community. Take a grid, and wherever there's a grid square in here, put a single cell in, and whatever the grid is covering, make that cell do that function. So if a grid is over here, then the cell that ends up being this part will take care of respiration. If the grid's over here, a cell that becomes that part will take care of reproduction, etc. And what do you end up doing is you start creating the model of a human embryo, which is this structure, and guess what? Has the same layers as the single cell. The outer layer of the skin is called the ectoderm, which is the skin and brain of the human embryo. The inner membranous structures form the inner membranous structures in our gut, respiratory, digestive, excretory system. The pink stuff is the spaces in between, and they form the mesoderm. What's the point? We are made in the exact same image, repeated again and again, and we now are cells in a larger organism. Each one of us has a job to support the life of the whole organism, so we are like cells in a multicellular structure, repeating it. This then leads me to this wonderful conclusion. It says, yes, civilization is a living system comprised of us as cells. And since the nature of fractals is self-similarity, it would say that then as human civilization evolves, it will have self-similar characters to the evolution of animals. So you can look at the status of a civilization and see where it is on the scale of evolution. So we go back and say, look, just looking at the vertebrates, fish, amphibians, reptiles, birds, and mammals. So where are we? Well, first of all, the fish cultures were in human evolution was called mariculture. These were the first civilizations we now recognize were on the planet. They lived at the edge of all the continents. They traveled across the water, continent to continent. They had to live with the water. Water, if there was water, they could live wherever that water was. But the next evolution was amphibians. Amphibians were unique. They took the water with them to the land. They didn't have to depend on being in the water anymore. They could take water with them. So the reality is, the amphibian phase of human civilization was agriculture, where we no longer were restricted to the water at the periphery, but using viaducts and irrigation systems, move into the land and populate the land, as the amphibians did. After the agriculture period, or amphibian period, come reptiles. Well, reptiles, if you ever see like these little reptiles, they're like little digital organisms. They click their head, their tongues flick, their eyes click, click like this. And you watch them and they're almost like machines. And in fact, dinosaurs are the fullest machine nature of a reptile. Here's why. A dinosaur and a lizard are exactly the same except scale. And when I was a kid, before animation in Jurassic Park, they made dinosaur movies like 100 million years BC or something. Victor Mature, God, that's a million years ago. And what did they have? They had dinosaurs. But what were dinosaurs? They took these little tiny lizard and glued some horns on them and stuff like that, and they photographed them real close, and God, they look like dinosaurs. And the fact is, they are. And dinosaurs are called machines. And the interesting thing about it, but here's where evolution went astray, because when the environment was perfectly stable and lush like it was in that period, then the evolution changed the dynamic, because evolution was expanding awareness. But at this point, when everything was stable, guess what? You could take a 5-inch lizard as a model. Let me give you an example. 5-inch lizard. The hind leg makes a movement like this. And I say, well, how many muscles does it take to move that? So I say 10 muscles. How many nerves does it take to fire those 10 muscles? 1. Okay. Now you take that 5-inch lizard and you make it 50 feet. Now the leg is this big. And it can still do this. Well, how many muscles does it take? 10 million to move that leg. How many nerves does it take? One. And that's why the dinosaurs grew bigger and bigger and bigger, and their brains were these little tiny things. Why? Same model, just played them bigger again. But the problem was, if the environment changed, there wasn't enough awareness in that brain to run the machine, change the dynamics. As long as everything stayed the same, you could run it exactly the same. But when an evolutionary change occurred in the environment, their awareness wasn't big enough to handle the system. They died out. But the interesting thing about that is awareness and body have a relationship. You know what they are? Awareness is brain, brain awareness, body is administration. What is administration? It handles the details of what the brain says. So that what happened in the dinosaur period was an imbalance started to occur, where the awareness stayed small, but the administration got bigger and bigger and bigger. Well, it's just like the world we live in. Government, healthcare, education, every one of these has small awareness and massive administration. We are in a dinosaur period right now. But there's some more relevant. What is the fuel that fuels our civilization today? The fossil fuel. The character of the dinosaur period is fueling our civilization. And the head of the country is what? The head dinosaur. He represents the oil company. Okay? Now, I'm going to come back to this in a minute because there's an interesting character to it, but let's talk about the evolution of the next two phases. So, what are reptiles? Reptiles is when the civilization hit the industrial age, the machine age. But then birds came from that machine age. Yeah. Does anybody remember when our bird civilization started? Wilbur and Orville Wright, 100 years ago. That change in evolution, this airplane changed the entire planet. Because no longer was it a forever distant place. You can connect anywhere. So we went from the industrial age, we entered the age of aviation and changed civilization on this planet. So now I ask you, what was the fullest extent of the bird phase of human civilization? The fullest evolution, what happened? We landed on the moon. And you know what happened? They sent back an icon that changed civilization. It was the picture of the Earth on the moon. It's behind this picture. And why did it change civilization? Mainly people like you in this room are the changes. It represented something when you looked at that and you said, oh my God, that's all there is. That's all we have, is that little thing. And all of a sudden the idea said, maybe we better take care of that place. Maybe we better take care of that water. Maybe we better look after that air. Maybe we better not pollute the ground anymore, because we'll destroy our own habitat. And that is called taking care of, is nurturing. Nurturers are what animal? Mammals are nurturers. And that's where we are now. But we're not all the way there, because there was a time where there were dinosaurs, mammals, and birds at the same time. And what happened was there was a crash of the dinosaurs. And the meek, the mammals, took over. We are the meek. And the difference between reptiles and mammals is reptiles are conscious and mammals are self-conscious. Meaning, reptiles live for the moment. They don't see the future. They live for what's happening right here and right now. Mammals plan what they do now to live in the future. And now George Bush may not be a willing participant. He just happens to be the person that's at the head of the show as doing what? The reptile. And to give you the consciousness of that reptile, I need not give you any better example than the war in Iraq. Why? Because they had enough awareness to get us into the war, and they couldn't even think 6 weeks down the road when the war was over. That is reptilian, living for the moment and dealing with the next moment when it shows up. That will be our end and our destruction. But the reality is this: they will fall. And then you have a choice. Your choice is to be on the mammal side or be on the dinosaur side. And why that's important is it's going to hurt a hell of a lot more if you're on the dinosaur side. Because the changes that are occurring will not support the world in this way any longer. Then until we come to the realization that we must take care of ourselves, our planet, we will not survive on this planet. This extinction that we're going through is a representation of that character. So there's an opportunity to make changes anytime. And the idea for you to be here is you've already started making changes. You want to know more about yourself and how you involve yourselves with others on this planet and how we can improve the planet. So you are the meek, but our time will come. And the idea is keep doing what you're doing because it will be our survival. We will not survive with reptilian thinking. It is totally destructive, and this one is coming whether you want to see it or not. Why? Evolution is fractal in its imagery. And I know I just went overtime a couple of minutes. Oh, I got 10 minutes. Oh, I got some great slides. The last 10 minutes, hold on. What's the evolution of this? I'll tell you what the evolution is. When it's fully evolved, it's the cell. The earth is a cell. And you and I are proteins in the membrane of the cell. We are perception units in the membrane, the skin of the planet. We receive awareness, and we turn that into behavior and life on this planet. We start to see the image right, the planet will survive. But if we do it wrong, it's pathology, and we're not going to make it. Now, the last closing slides are this. All the people out here in the room are individuals. Every one of you is an individual person. You have your own name, your own identity, but you have something more than that. Your cells know you and have your brand on them, and nobody else has that brand. If I take your cells and put it into the person sitting next to you, what's going to happen to those cells? They'll be rejected by the immune system of the recipient. For what reason? They're called not self. So all of a sudden they say, where's the self? What is the self? What makes you, you, and the person next to you not you? Protein, so I'm going to get to it. I use 2 cells, Bruce's and Margaret's. I use 2 liver cells because 99% of the cell is liver in function. 99% of the cell is exactly the same as Margaret and Bruce. They're both the same. Liver cells do the same thing. Liver enzymes in me would be the same as the liver enzymes in Margaret. Same thing. I know the nuclei are different because we have different genes, but I also told you this. I could throw the nuclei out of the cell, and the cell's still going to have a life. So I'm not going to count them in this story right now. What I'm going to do is remove the color from the picture, leaving behind the color where the 2 cells differ from each other, where the identity is found. Where is it that makes us unique? And I remove the color. Can you see where the colors are left? They're in the membrane. And biologists even call some of these self-receptors. What are receptors? Receivers. What are self-receptors? Receivers of self. And why is this relevant? If I take my self-receptors off my cell, I take an enzyme and cut off these yellow receptors, you know what happens? My cell is generic. I can implant it in any human, it will never be rejected. I can implant it into a mouse, it won't be rejected. There's no identity to it. So I take my generic, no identity cell, my cell, and I take Margaret's receptors off of her cell and put it onto my cell. So I have my cell with Margaret's receptors. If I put that cell back in my body, what's going to happen? It'll be rejected, not self. If I take my cell with her receptors and put it into her body, what's going to happen? It's accepted. I transfer ownership by just transferring the antennas. Now you might think, well, these antennas are the self. The fact is, antennas are antennas. They are receivers of a signal, so that your antennas complement a signal out here in the environment. Why would I say that? Because the receptors are reading the external environment. They're not reading the internal environment. So why is that relevant? Because your identity, these receptors on the surface, are reading an environmental signal that's unique to you. And if I take that signal and transfer it to somebody else's cell, then that cell becomes your cell because that signal is going to control the cell. The signals that come in from the outside give it the identity. And the relevance about that is that, let's just say, I'll start with this first, give you another story about it. If I take your receptors and I put it on an embryo, and I let the embryo develop with those receptors, who is that individual? It's you. But did I say, was it a male cell or a female cell? Did I say it was a white human or a black human? Well, that wasn't relevant. The identity and the body are two different things. One controls the body. What does this mean? It says that in a future date, if an embryo shows up and just happens to have the same set of receptors on its surface that you express now, you're back online again. That you've never died. What do you mean I never died? Well, I'll give you an example. As of late, because of the large number of transplant operations, heart-lung combinations, when people receive heart-lungs from other people, they don't just receive the organ, they begin to acquire the character of the person who donated the organ. Here's a fact that's so amazing. A young girl got murdered. Her heart was transferred to another person. That other person was able to identify the murderer for the police. It lived and knows. What was the point? You'd say, oh, the cells are carrying the memory. I'm going to tell you something. I'm a cellular biologist. The brain is cells, that membrane. It does not have the awareness capacity to distinguish chicken McNuggets and beer from anything else in the world. It doesn't do that. The memory is not in the cell. The memory is in the field. We are a broadcast. This is a television set. We have antennas. When the picture tube breaks on your television, what do you say about the TV? The TV is dead. Yeah, TV dead. But did the broadcast stop? No. Well, how can you tell that? You get another TV set, you plug it in, you turn it on, you do one other thing. What do? Tune it to the station. And when you hit that frequency, boom, it's back on again. That we are immortal. We don't even live in this system. This is a system for us to come into and then live out life. And then you might say, well, Bruce, which I ask my own self, I say, Bruce, if I live out there, then why live in here? Why this? And my cells gave me an answer. And my cells said to me this, Bruce, if you're just a spirit, what does chocolate taste like? What does a sunset look like? What does being in love feel like? Why is that relevant? The answer is, it's the cells that take that environmental information and convert it into an awareness that I can understand. That the real world out here is converted into electromagnetic vibrations. I'm reading the energy. I'm not reading the chemistry. I'm not physically seeing the light. I'm reading the energy of that. So my physical body is a device to sense the world. It's a device to come in here to create a world. That if anybody has the fondest idea of a heaven and they're looking elsewhere, I think it's a great mistake. Because your opportunity was to come here to create what you thought you would create in heaven. And because it looks like hell, that's because you bought other people's creations. And that you have the opportunity to take over your own life and create your own life. And I didn't know that as a cellular biologist who was teaching biochemistry and genes. But when I owned it, and I caught a hold of those beliefs that I acquired in the first few years, the limiting beliefs. The one that said you can't heal yourself. The one that said you can't do this, you're not smart enough, you're not good enough, whatever those things are, we all heard, and then we become. If you eliminate those and have a blank slate and create what you want, you can create anything on this planet, including that dream you thought heaven was. It's your dream, you can create it here. And the lesson is, we let go of this physical reality and own who you really are. Who are you? Well, the simple reality is this. You know those receptors? Let's say that they're like, let's say the membrane's opaque. Let's say the membrane is opaque, nothing gets in. So what color is on the inside if nothing gets in? Black. But now I say, my self-receptors are yellow, so it's like wearing a yellow sunglasses. Margaret's self-receptors over there are red. She's wearing a red sunglasses. And the light can come through the membrane, so what color is on the inside of mine? And on hers, red. And what's the point? Where did the color come from? Where did the color come from? From out here. All that is. And what does it represent? I am just a narrow frequency of the whole white light. You are a narrow frequency of the white light. You are a narrow frequency of the white light. Every one of you. What does it mean? To me as a biologist, it means this. When people talk about the white light coming back to the planet, they generally envision some person like a Jesus coming back. I think that's the biggest error of all time. The white light is here. The white light is when you put all of the frequencies together, you have white light. If you remove one solitary single frequency from the white light, you do not have white light. When we take a person and say this person doesn't fit and doesn't belong, and you throw them out, you cannot have white light. The biggest lesson is to find out we are one living organism called humanity. We are all the cells in that organism, that every human is a cell in that organism, but their educations are not correct. And it's readjusting the education and letting go of all the history in the past that made limitations and self-sabotaging beliefs. It's forgiving and letting go and starting with a white blank page saying, what do you want from here on, not who did what to who back then. And this from a biologist who didn't know a damn thing about life until this understanding of the membrane hit me. And the reality is, I sit here in awe. Every day I wake up in awe to be on this planet. And if you're not seeing that, then it's time for readjusting the glasses. It's time to see it again. Because all of you, the person sitting next to you, the person sitting behind you, are part of this white light. And as a biologist, all I can say is, man, we have really wasted a lot of time. And yet the heaven is when we walk out this door, it's available to you right now. Thank you very much for your listening. I appreciate it very much. Thank you. Thank you very much. Thank you. Thank you. Thank you very much.